Hello. So, very good afternoon to the student who have joined. Can you please let me know if my voice is audible? And also, please let me know if you can see the video and also the presentation. Let me know if it is audible and visible too. Okay, so the presentation is visible, right? Okay, thank you for confirming. We will wait till more students join and then we will start today's session. Meanwhile, please wait and also let me know if there is an issue with the audio and the presentation. Okay, so it's 4 p.m. Let's start today's class. Okay, so very good afternoon to the students who have joined. My name is Moshumi Dev. I'm a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. Today it is our week 8 live session for the NPTEL course of Medicinal Chemistry. So every week we solve a particular week's course content via the MCQ questions in the live session. So I will today also I will give you around 20 MCQ questions, and all the questions will be based on the week 8 course content of this medicinal chemistry course. Okay, each question will have four different options as answers, and there will be only one correct answer which you have to choose. But before starting the session, I would like to point out few things. First, like there will be this type of problem solving sessions every week. Okay. And the sessions will be on every Friday from 4 to 6 p.m. The schedule is same for every class. It is Friday from 4 to 6 p.m. and the sessions are live on Google Meet platform, which has this particular link. The link is same for every class, so you can just save it somewhere and use it every Friday to join every Friday session. So very important thing is if there is any network disruption from my side, please do not leave the session immediately. Wait, I will rejoin as soon as possible. And during the session, the students can post your doubts and queries through the chat box. Since I'm recording the session, okay, so that because of that, I have muted your microphones and cameras so that I can record the session seamlessly. But the chat box is always open. I'm monitoring the chat box every time. So if you have any problem, any doubt, any query regarding the class, regarding the content or anything, feel free to let me know in the chat box. Okay, I will try to answer your queries during the class itself, or at the end of the session I will share my email ID with you also. So if you have any doubts, any query which are not answered during the session, you can just drop an email regarding those things, and I will answer your query via email also. Okay, and as I have already mentioned, I'm the recording the class and the presentation which I am presenting today. These things I will be sharing with you. And somebody also has asked me where to find this problem solving session. I have actually shown you earlier also. So just have to go to the SWAYAM portal, where is the course content page. Okay, so you will have a thing which is the announcement. So it is the announcement section. You will find there is a at the left panel, you will find a problem solving session January 2025. If you click on this position, you will be redirected to a page. Okay, here, if you click on this page, you will find the recording and the presentation of the problem solving session. Okay, so somebody has emailed me regarding that, so I thought to share you once more time. Although I have showed you many time, but it's fine. So you will be able to access the presentation and the recording via that particular link. Okay. So, as I have already mentioned, today it is our week 8 live session, and what do we will do today? We will discuss the week 8 course content. Okay. So, like every session, I will give you the MCQ questions. Every question will have four different options, and you have to choose which of the following you think is the correct answer, and let me know in the chat box. After I will check the chat box and I will show you the correct answer. And after that, what we will do is we will learn about that particular topic, or we will discuss that particular topic in detail. Okay, so there is no negative marking, or there is no kind of marking because for answering during the class. So you can answer fearlessly. Your answer, even if it is wrong, it won't be it won't be marked. So there is no problem. But answering during the class is very important. Otherwise, I won't understand if you are following the class or not. So let us first see what is the week 8 course content. So last class in week 7, what we had discussed, we basically are discussing right now the different aspects of the ADME, right? Your administration part, the drug administration, what are the different routes of administration, what is viability, and all those things we have discussed in our previous session. Okay, we have discussed several aspects of pharmacokinetics as well in the previous session. Today, the main topic which we are going to discuss is the optimization of drug target interaction. Okay, designing the drug changing some modification or doing modification in that particular drug structure to enhance its effect that with the target or enhance its interaction with the target. These things what are we going to discuss in today's class. Okay, so basically we course contents are the optimization of drug target interactions, which are divided into four different parts with different topics. Okay, so let us start. So after every question, I will wait for some time and you have to pick up one option as a correct answer. Okay, so let us see what is the first question of today's session. So the first question of today's session is, which of the following statements best describes the structure activity relationships or SAR? So your options are number A. It is the study of which functional groups are important to the chemical reactivity of the drug. Number B. The study of the physiochemical properties that are important to absorption of the drug into the blood supply. Number C. The study of the structural features of the drug that are important to its biological activity. And number D. The study of the structural features of a drug that are important to its chemical stability. Okay. So you have to find out A, B, C or D which of the following statement is correct regarding SAR or structure activity relationship. Again, I will repeat the options. Okay, it is the study of which functional groups are important to the chemical reactivity of the drug. Number B, the study of the physiochemical properties that are important to the absorption of the drug into the blood supply. Number C, the study of the structural features of the drug that is important to its biological activity. Number D, the study of the structural features of the drug that are important to its chemical stability. Which of the following is the correct answer? Which of the following statements best describes the SAR? I will wait for 1 or 2 minutes, then I will check the chat box. Okay. Okay, let me check if there is any answer in the chat box or not. Okay, I have received two answers. Both are saying it is number C. Okay, let me check if number C is the correct answer for this question. Yes, number C is the correct answer. SAR basically is the study of the structural features of the drug that are important to its biological activity. Now let us discuss about SAR a little bit. So basically, structure activity relationships. So once the structure of the lead compound is known, lead compound, what is a lead compound? How to figure out or how to find out a lead compound for a particular disease or drug target, or what are the requirements of a lead compound? All these things I have discussed in the previous sessions. Okay, so you can go through the PPT if you do not remember or if you have any doubt regarding those things. So once the structure of the lead compound is known, the medicinal chemist moves to the study of its structure activity relationships or SAR. Okay, the aim is to identify those parts of the molecule that are important to biological activity and those that are not. If it is possible to crystallize the target with the lead compound bound to the binding site, the crystal structure of the complex could be solved by the X-ray crystallography, then studied with the molecular modeling software to identify the important binding interactions. However, this is not possible if the target structure has not been identified or cannot be crystallized. It is then necessary to revert the traditional method of synthesizing a selected number of revert to the traditional method of synthesizing a selected number of compound that varies slightly from the original structure, then studying what effect it has on the biological activity. One can imagine the drug as a chemical knight entering the body in order to make the battle with an affliction. And then the drug is armed with variety of weapons and armor, but it may not be obvious which weapons are important to the drug's activity or which armor is essential to its survival. What we can find out only by removing some of the weapons and armor to see if the drug is still effective. The weapons and armor involved at a various structural features of the drug that can either act as binding groups of the targeting site or to assist or protect the drug from its journey in the body. Recognizing functional group and sort the intermolecular bonds that can form an important is an understanding is how the drug might bind to the target. Okay, so this thing which is has been beautifully describes this SAR is that the drug, every chemical moiety has some structural features. Okay, for example, let us say we have benzene, we have chlorobenzene, we have toluene. Okay, so let's say it is a lead compound. So now the change from the benzene to chlorobenzene to toluene is like only a one functional group is vanished out, right? So there is either chlorine or methyl or there is hydrogen. So if that is a lead compound and we have to check that which of the particular compound is active for a particular target, and this also give us the indication that a particular group or particular functional group in that particular drug is important for the binding, either binding to the target or for the drug stability in the body. So these things are studied in the SAR or structural activity relationships. So basically, this is an example, okay, for this compound. So structure activity relationship, which means structure determines the function, is presented by Crum and Brown and Fisher by in 1865. The structure of the compound determines its biological effect. Biological effects it have. Determination of the structure and function relationship helps the medicinal chemist to synthesize new drugs and pharmaceutical agents. So, for example, this is a compound. Let us say this is a lead compound, and here each and every group or each and every bond has a particular role. For example, this methoxy group is important for the binding stability. This methoxy group is not required at all, and this methoxy group is important for the correct conformation or activity. Here we also have a methoxy which permits the halide, alkyl, alkoxy amino acid and its methyl group but not bulky group. So this position we can change this OMe from OMe to halide, alkyl, alkoxy and so on. This allows this particular bond, this allows the ring contraction. This group can be replaced by other substituents. Okay, and epimer diminish the activity. Here we also see there is a, I mean the this has a chiral center which has a particular epimer, a particular enantiomer. Okay, this particular bond is important. Okay, ring expansion is not permitted because of this bond, and unsaturation increases the activity here. And this is the particular SAR or structure activity relationship with the different structural features and with the particular drugs activity and stability. Okay, so this is all about the structure activity relationships. We will see more about this SAR in our upcoming questions also and upcoming sessions also. So this was the basic of what is SAR and how it works. Now let me move to the next problem. Problem number 2 says SAR shows that the important binding groups in morphine, in binding group in morpholin are phenol, aromatic ring, and amine. A diagram can be drawn which shows these functional groups and their relative or orientation. What is this term for this? Your options are number A pharmacodynamics, number B pharmacokinetics, number C chromophore, number D pharmacophore. Again, I am repeating the question. SAR shows that the important binding groups in morpholin are phenol, aromatic ring, and amine. A diagram can be drawn which shows these functional groups and their relative orientations. What is the term for this particular diagram? Your options are pharmacodynamics, pharmacokinetics, chromophore, or pharmacophore. A, B, C, or D, which of the following will be the correct answer? A diagram can be drawn with the different functional group and their relative orientations, which are important binding groups. Okay, what is this term form for this diagram? Pharmacokinetics, pharmacodynamics, chromophore, or pharmacophore? A, B, C, or D? Let me know in the chat box A, B, C, or D which of the following is the correct answer. Okay, let me check the chat box. Okay, so I have, I think, two options. Both are saying it is number D. Okay, let me check if number D is the correct answer or not. So for problem number two, the correct answer will be yes, it's the number D, pharmacophore. So now let us see what are the all these terms that depict. So basically, pharmacokinetics, I have already discussed it in for quite a few times that it is how a particular drug, how our body, how our body is doing to a particular drug. Okay, so how a drug is we are taking, our body is taking the drug, it is distributing the active components of the drug in our body, it is metabolizing that particular things and excreting. Okay, so there is the pharmacokinetics, the ADME. Okay, this is the quantitative study of the drug movement in, through, and out of the body. It is the study of the process by which the drug is absorbed, distributed, metabolized, and eliminated in the body. The absorption, distribution, metabolism, and excretion of the drug all involve its passage across the cell membrane. The pharmacokinetic parameter properties are affected by the route of administration and the dose of the administered drug. Pharmacodynamics is the study or mechanism of the action of the drug and explains what drug does to the body and how they do it. It tries to explain the action effect relation as well as the dose relation effect. It involves the drug receptor interaction to elect the action of the drug in the body. So basically, the pharmacodynamics is what the drug is doing to our body, how it is affecting our body, but when it is interacting with different targets. The pharmacodynamics is the study of the biochemical and physiological effect of the drugs and the metabolism in action. It provides the basis for the rational use of the drugs and the design of the better and effective drugs. Then we have chromophore. Chromophore is a Greek word, okay, where chroma means color and phore means the bearer. It is defined as any isolated covalently bonded group that shows a characteristic absorption of the electromagnetic radiation of the UV or visible region. For example, compound having chromophore is called the chromogen, like C11C, C11O nitro. These are the chromophore. Then we have pharmacophore. So what is the IUPAC definition? It's an ensemble of the steric and electronic features that is necessary to ensure the optical supramolecular interaction with a specific biological target and to trigger or block the biological response. Okay, we have hydrogen bond donor, hydrogen bond acceptor, and these are the all the depiction. So in drug design, the term pharmacophore refers to set of features that are common to a series of the active molecules. Hydrogen bond donors, acceptor, positively and negatively charged group, and the hydrophobic regions are the typical features. We will refer to such features as the pharmacophoric groups. So basically, a pharmacophore concept was proposed by Paul Ehrlich in 1909. Pharmacophore is defined as a molecular framework that carries the vital features required for the biological activity of the drug. Okay, and IUPAC definition: pharmacophore is an ensemble of steric and electronic features that is necessary to ensure the optimal supramolecular interaction with a specific biological target or to trigger or to block its biological response. The pharmacophore model is uses to express the 2D and 3D images of the molecule and to depict the key elements of necessary binding. This concept was generally used for binding to identify the target, virtual screening, molecular docking and simulation, etc. A pharmacophore model explains how structurally diverse ligand can bind to a common receptor site. Okay, so this is all about the pharmacophore. So option number D was the correct answer. Now I will go to the next problem. So problem number 3 says combristatin is an anti-cancer agent discovered from an African plant. Analog 1 is more active than the combristatin, whereas analog 2 has less activity. Which strategy is used in designing the analog 1 and 2? So this is the combristatin, okay, and this is the analog 1 and analog 2. The analog 1 is more effective, whereas analog 2 is less active. So which strategy is used to design the analogs 1 and 2? From this to this and this, which strategy has been used? Your options are number A, simplification, number B, isosteric replacement, number C, rigidification, number D, chain contraction. Which of the following strategy is used for designing the analogs 1 and 2 of the combristatin, which is a very potent anti-cancer agent? Simplification, isosteric replacement, rigidification, or chain contraction? A, B, C, or D? Let me know which of the following is the correct answer. Combristatin is an anti-cancer agent discovered from African plant. Which strategy is used in designing the analog 1 and 2? Let me check the chat box. Okay, I have one answer which is saying number B and another saying it is number C. The two different options I have received as answer. Let me check which of the following will be the correct answer. Okay, so the correct answer is it is number C, rigidification. So now why? Let us first, I may just show you one thing. If you notice that this is the parent structure, right? So there is a single bond is here. This bond is single, which means this particular entity, this bond, the rotation is possible around this bond means at this position. But when we introduce a double bond here, okay, means we are restricting the structure or we are making this structure rigid. Now this bond is not rotation, the rotation is not possible around this bond anymore. Here it is this also same thing. This is the same isomer. One is the cis and another is the trans, and these structures are rigid. Okay, rather than this parent structure when there is no double bond here, because of that this molecule is much flexible for the rotation, but these molecules are rigid. That is why this particular I mean design strategy is known as the rigidification. So now first let us understand what is the rigidification strategy. So rigidification is often used to increase the activity of the drug or to reduce its side effect. In order to understand why this tactic can work, let us consider the, okay, so there is a hypothetical example. So let us say this is a neurotransmitter, okay, which is quite flexible. As you can see, this particular group is quite flexible, and flexible molecule with a several retroshaban ball that can lead to large number of conformations or shape. Okay, so if a molecule is flexible and there is a several bonds that can rotate, so what will happen? This particular structure will lead to a large number of conformations or shapes, and one of these conformations can be recognized by the receptor but is not known as the active conformation. The other conformations are unable to interact efficiently with the receptors and the inactive conformations. However, it is possible that a different receptor exists and which is capable of binding one of these alternative conformations. If it is the case, then our model neurotransmitter could switch on to different receptors and give two different biological response, one of which is desired and one of which is not. Okay, so basically, if a structure has given to rise to different conformation, what can happen? One conformation is our desired, which will activate a particular desired receptor to get a desired biological effect. But it may happen that the different conformations which are raised from that particular parent compound, that particular conformation can activate or it can block other receptors, so giving rise to the side effect, which is not what we want. So the strategy of rigidification is to make the molecule more rigid, such as the active conformation is retained and the number of the other possible conformation is decreased. This should reduce the possibility of other receptor interaction and the side effect. This same strategy should also increase the activity by making the drug more rigid. It is more likely to be an active conformation when it approaches the target binding site and should bind more readily. This is also important when it comes to the thermodynamics of the binding. A flexible molecule has to adopt a single active conformation in order to bind to this target, which means it has to become more ordered. This results with a decrease of entropy, and as the free energy of the binding is related to the entropy by equation delta G equal to delta H minus T delta S. Any decrease in entropy will lead to an effect to increase the delta G. In turn, this lowers the binding affinity or the Ki. K1 which is related to the delta G by the equation delta G equal to minus RT ln K. A totally rigid molecule, however, is already in the active conformation, and there is no loss in entropy involved in the binding with the target. If the binding interaction delta G are exactly the same as the flexible molecule, the rigid molecule will have overall better binding affinity. Okay, so this is how the rigidification is supported by the thermodynamics also. So for example, here you can see this is a flexible messenger, a flexible neurotransmitter, let's say, which has the active conformation. These bonds are rotatable bonds which can adopt this conformation also. Then this particular conformation is not active. Okay, but if we do the rigidification, means if we introduce a particular type of ring here, so what will happen? This ring will attain this active conformation, but it will not now be possible for this molecule to adopt this kind of conformation. So that is how the rigidification works. So what is the rigidification? This is a strategy which introduces rigidity to the particular active compound by this thing. What how it affects basically? This will lower the it will lower the number of active conformations, and any kind of undesired side effect will be diminished. Okay, so this was the problem number 3. Now I will go to the next problem. Problem number 4 says, what is the reason for the improved activity of the compound B rather than the compound A? What is the reason? Okay, there is a two compound, both have the exactly same structure, except one thing, that there is no nitrogen here, but there is a nitrogen basically. Okay, so this is the thing. So now you have to identify why this compound has better activity than this compound. Your options are: number A, less toxic metabolite formation; number B, increased solubility; number C, increased bioavailability; number D, good pharmacophoric profile. Which of the following will be the correct reason for the improved activity of B compared to the A? I want all of you to answer the question. Even if your answer is wrong, it does not matter, since we are not going to mark your answer in the problem solving session. But answering the questions is very important. So just try A, B, C, or D. Which of the following you think is the correct answer? So here we have a indole ring, as you can see in this molecule, and in this molecule we have a aza-indole ring, but otherwise all the things are same. But this compound B has the improved activity than A. What is the reason? Number A, less toxic metabolite formation; number B, increased solubility; number C, increased bioavailability; number D, good pharmacophoric profile. Okay, let me check the chat box. Okay, I have few answers. One is saying it is number B, and other two answers I have received which are saying it is number D. Okay, let me check which of the following is the correct answer for problem number 4. So the correct answer will be number A, less toxic metabolite formation. So this compound, this parent compound, so basically it is a HIV-1 attachment inhibitor or anti-HIV drug we can also say. So basically this compound which has a indole ring, so this ring has a methoxy group in para position. Whenever there is this kind of methoxy group in para position, during the metabolization, what happens? These methoxy groups are converted to the phenolic OH group, and then after that they give rise to this kind of quinone ring, and this quinone ring is very toxic. It is also known as the toxicophore. Okay, this quinone ring, whenever this is a metabolization happen, so they, because of the, so whenever there is we are introducing this particular nitrogen here, two things happen. One, the soluble aqueous solubility of the drug increases, and another thing is because of this the introduction of this nitrogen here, now this particular metabolite is formed, which is the non-toxic amide. Okay, and this is also, this is the and this is the metabolization which happens in vivo, means when this particular compound is tested in the mice or any kind of animal model, then this type of metabolization are noticed. Okay, that is why during the development of this particular drug, which is the very important candidate BMS 488043, which is in the phase 3 clinical trial, this nitrogen is introduced here, when which is which is found that this compound has a better activity, and the reason is the increasing, a little bit of increasing the aqueous solubility, but the diminishing the toxic effect also. Okay, and so this less toxic metabolite is the main reason why the activity of B is greater than the activity of A. Okay, so the correct answer was problem number A. Sorry, the option number A. Now I will go to the next problem. Problem number 5 says, which of the following strategies was used to develop the anti-cancer drug crizotinib? Your options are number A, ring fusion, number B, ring cleavage, number C, chain contraction, number D, all of the above. Which was of the following strategies was used to develop anti-cancer drug crizotinib? Ring fusion, ring cleavage, chain contraction, or all of these techniques are used for developing this particular drug? A, B, C, or D, let me know which of the following is the correct answer. The strategy or strategies used to develop the anti-cancer drug crizotinib. Which of the following strategies was used to develop anti-cancer drug crizotinib? Let me check the chat box now. Okay, I have. So I think I have got only one answer, which is saying it is number D. Let me check if number D is the correct answer or it is not. So the correct answer will be yes, it is number D. All of the above strategies were used to develop this anti-cancer drug crizotinib. So basically, this is the example of the structure-based drug design. Okay, so structure-based drug design is normally used to observe the binding interaction of the ligand and then identify the modification that will result in the better interaction and greater activity. This approach was used to design recently approved anti-cancer drug crizotinib and included in the substantial modification which is totally altered the scaffold of the molecule. PHA 665752 was the starting point. Okay, so this is this molecule as you can see here. This molecule PHA 665752 is the starting point of this particular structure-based drug design approach of the drug approach. So this had been obtained from a structure-based drug design of the previous lead compound. However, it had a large molecular weight and was too hydrophobic to be orally active. So we all know that there is a rule which we have to follow, the Lipinski's rule, which has a particular requirement of the molecular weight, lipophilicity, and so on. So particularly this molecule was not fit for those criteria, and that is why this molecule was not very good, very potent, and the structure was co-crystallized with the target enzyme, and the crucial binding interaction were identified. These included the hydroquinone ring system. Okay, and this is the hydroquinone ring system. The dihydroindolone ring system was found to be important with the hydrogen bonds, hydrogen bond donor and hydrogen bond acceptor. Okay, so this particular ring is important because it has a hydrogen bond acceptor and hydrogen bond donor. Okay, and as well as the dichloroaromatic ring. So this one, this dichloroaromatic ring is also important. As a result of the study, it was noted that much of the scaffold connecting these binding groups were reducted and so much simpler, less hydrophobic skeleton was designed, which has a position important binding groups in a similar way, but in more fashion manner. The thought process behind this design involved a ring fusion, ring cleavage, and chain contraction. When the novel structures were synthesized, they were found to bind as predicted, and was further structure-based drug design was used to optimize the process to leading to the crizotinib. So first, this molecule, as I have already mentioned, this dihydroindolone group is important because it has a hydrogen bond donor acceptor. So thought process is first one was a ring fusion to minimize the number of connecting rings or so. So we have this compound. This is the first thing, and then thought process two was the ring cleavage. Ring cleavage. Why the ring? So ring cleavage is cleaving this particular ring, so this particular position, because we have to retain this hydrogen bond donor and hydrogen bond acceptor. So as you can see, this ketone ring here acts as a hydrogen bond acceptor, which is modified to this pyridine nitrogen, which is also a hydrogen bond acceptor. And in the second thought process, when the ring cleavage happens, we get this NH2 group here rather than this NH, which is the hydrogen bond donor is also is there. Then we have the thought process 3, which is the chain contraction. Okay, and the chain contraction, what does it do? Is simply changes this our particular this remove this particular chain and this group to the simplest one, and others optimizations, more optimizations give us this particular drug crizotinib, which has this particular dichloroaromatic ring, which is important, and also ring aromatic center ring, which has a hydrogen bond acceptor and hydrogen bond donor. Okay, so this was the correct answer. And that is how basically the structure based drug discovery happens. So this is a very excellent example of the structure based drug discovery. Okay. Now I will go to the next problem. So problem number 6 says succinylproline was the lead compound for captopril, which acts as an inhibitor of the angiotensin converting enzyme. What is the relevance of this thiol group? Number A, it can form a covalent disulfide bond with the cysteine thiol groups in the active site. Number B, it acts as an isostar of the carboxylic acid. Number D, it acts as a bioisostar of the carboxylic acid. Number D, it has a smaller group than the carboxylic acid and fits the binding site easily. So ACE, angiotensin converting enzyme, is a very important enzyme which we have already shown in the in the renin angiotensin system. And the inhibitors of ACE, what does they do? They are basically very good antihypertensive drugs, right? So this is one very important example is captopril, which is the ACE inhibitor, and succinylcholine was the lead compound for this development of captopril. Okay, so there is this thiol group in the structure of the captopril. You have to identify what is the relevance of that particular thiol group. Your options are number A, it can form a covalent disulfide bond with the cysteine thiol groups in the active site. Number B, it acts as an isoster of the carboxylic acid. Number C, it acts as a bioisoster for the carboxylic acid. Number D, it is a smaller group than the carboxylic acids and fits in the binding site more easily. Let me know A, B, C or D, which of the following is the correct answer. All of you try to answer the question in the chat box. After a minute, I will check the chat box and then I will discuss this thing. Okay, let me check the chat box. Okay. So I think I have three different options as answer. One B, one C, and one D. Okay, let me check which of the following is the correct answer. B, C, and D. We have three different answer. Let us check. The correct answer will be number C, it acts as a bioisoster of the carboxylic acid. Okay, so bioisosters is a very important concept. We will discuss it later. What are the bioisoster? What are the relevance of bioisosters? So we will discuss. So basically, let me just give you a brief about this thing. So sometimes what happen, a functional group which is very important for the activity of drug, but for some reason we cannot use that particular functional group as it is. Okay, so for example, carboxylic acid due to some metabolism problem or some up to high, I mean the polarity of the carboxylic group in the drug, we cannot use that carboxylic acid group as it is. Okay, we have to use some other group which has similar kind of activity of that carboxylic acid group, but not as exactly that particular group. So whenever the because it has the same biological activity. Okay, that is why it is called the bioisoster. Bioisoster mean this particular group will be having similar biological activity of that carboxylic acid group, but it will be more metabolically stable and it will not give rise to many kind of different kind of your side effects. So this is the succinylcholine which has the IC50 value of 628 micromolar. We have discussed what is IC50 value. It is the inhibitory. This is the concentration, the inhibitory concentration minimum inhibitory concentration to give rise to the 50% of inhibition. Okay, so 625 micromolar means this much of amount of concentrated drug is required to inhibiting this this AC in enzyme to the 50% inhibition, and then this is the intermediate group which has this which has IC50 value of 52 micromolar, and this is the captopril which is the final compound. So this captopril has IC50 value of 23 nanomolar. So lesser the IC50 value, more potent is the particular inhibitor molecule. Okay, because lesser amount drug is required to inhibit that particular enzyme's activity to 50%. So which are so captopril, as you can see here, succinylcholine we have a carboxylic acid group, but in the captopril we have a thiol group. So this thiol group is acting as the bioisoster of the carboxylic acid group in our lead molecule. So basically, isosters of or bioisters of carboxylic acid group. So it has different groups acts as an isostar, and like tetrazole, the hydroxylamines, then acetyl sulfonamide, sulfonic acid, phosphamide. So all these groups have a similar pKa as the carboxylic acid group. Okay, so not all these groups can be used as a bioisostar of carboxylic acid because all these groups will not give rise to similar biological effect that is different from the different for the particular structure. So these things we will discuss in later on also. So the option number C was the correct answer. Now I will go to the next problem. Problem number 7 says the procedure of transforming a racemic drugs to the single enantiomers is called dash. You have to fill in the blanks. Your options are number A chiral pool, number B asymmetric synthesis, number C chiral switch, number D asymmetric switch. The procedure of transforming racemic drugs to the single enantiomer is called dash. You have to fill in the blanks. Number A chiral pool, number B asymmetric synthesis, number C chiral switch, number D asymmetric switch. What is the correct answer? What is the name of the procedure which is used to transform racemic drugs to single enantiomer? A, B, C or D? Let me know. What is the procedure? Let me check the chat box. Okay, so I have two different answers. One is saying it is number C and another saying it is number B. Let me check which of the following is the correct answer. So the correct answer is yes, it is number C, chiral switch. Okay. So chiral drug, basically we have discussed the relevance of the chiral drug or the relevance of having a single enantiomer as a drug, and very good example of this thing is the thalidomide, which we have already discussed in our first its session itself, I think. So chiral drug is a single agent used instead of a mixture of two distinct drugs. Okay, which simplifies the interpretation of the basic pharmacology, greater selectivity for their biological targets, improved therapeutic indices, and reduced adverse effect, longer or shorter duration of action, and more appropriate for the dosing frequency. So chiral switch is the procedure to use to transform an old racemic drug into its single active enantiomer. The new enantiomeric drug developed by the pharmaceutical manufacturer will receive the additional patent protection and no generic name. Okay, so asymmetric synthesis is basically the synthesis of a single enantiomer rather than a mixture of enantiomer. That thing is different. Okay, so asymmetric synthesis is the procedure of synthesizing a single enantiomer itself, but the procedure of transforming the racemic drug to a single enantiomer is known as the chiral switch. Okay, so problem number answer. So C was the correct answer for problem number 7. Now I will go to the next problem. So problem number 8 says the use of one drug for activating different targets is known as dash. You have to fill in the blanks. Your options are number A additive therapy, number B combinatorial therapy, number C MT or multi-target therapy, and number D none of the above. The use of one drug for activating different targets is called dash. Additive therapy, combinatorial therapy, MT or multiple target therapy, and number D none of the above. Let me know A, B, C or D which of the following will be the correct answer. What is the name of this particular therapy? Let me check. Okay, I have got one answer which is saying number C and another is saying number B. Let me check which of the following is the correct answer. So the correct answer will be number C, MT therapy or multiple target therapy. So designing drug that are able to interact with the more than one target. So many diseases require a cocktail of drugs interacting with different targets to provide the suitable treatment. A better approach would be to design agents that interact with two or more targets in a controlled fashion in order to reduce the number of the drugs that had to be taken. This is known as the multiple target drug discovery or MTDD. So this is so one drug it is able to activate different target to have the maximum therapeutic effect. So there have been two approaches to design such a multi-target directed ligand. One is to design agents from the known drugs and pharmacophores such that the new agent has the combined properties of the drugs involved, and the other approach is to start from the lead compound which has activity against the wide range of the targets and then modify the structure to try and narrow the activity and narrow the activity down to the desired target. Okay, so these are the approaches for the designing the drug for this multi-target drug discovery. And what are the advantages of this MT therapy? So more predictable and superior pharmacokinetic and safety profiles as the consequence of administering a single drug. The combination of multiple active principles may result in positive but also negative synergistic effect, which limits the number of useful combinations. The probability of developing target-based resistance simultaneously to multi-target drugs is statistically lower than the probability of developing resistance against the single-target drugs. The risk of the drug-drug interaction associated with the combination therapies is avoided, and the therapeutic regimen is greatly simplified. The use of drug cocktails often complicates the dosing schedules and negatively impacts the patient compliance. It's even more difficult to perform the clinical trials with the drug combinations if the drugs come from the different companies, and higher efficacy against the complex diseases such as the cancer. Okay, so these are the advantages of the MT therapy or the multiple target therapy. Okay, so now I will go to the next problem. So problem number 9 says, by which method the chimeric drugs are formed? Your options are number A, in silico drug designing method, number B, pharmacophoric hybridization method, number C, bioisosteric replacement, and number D, structure based drug design. So by which method chimeric drugs are formed? Number A, in silico drug designing, number B, pharmacophoric hybridization, number C, bioisosteric replacement, number D, structure based drug design. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 9. By which method the chimeric drugs are formed? In silico drug designing, pharmacophoric hybridization, bioisosteric replacement, structure-based drug design. So, what is the correct method for designing the chimeric drugs? Let me check the chat box. I have received one answer only, which is saying it is number A. Others wants to try, or okay, let me check if I think I got one answer which is saying it is number A, right? Okay, let me check if number A is the correct answer or there is another correct answer. See, answering during the class is very important. Even if our answer is wrong, it does not matter, but it is very important to answer. So let me see which of the following will be the correct answer. So the correct answer is number B. Pharmacophoric hybridization is the method by which the chimeric drugs are formed. So what are the chimeric drugs? So another method is to, so basically the design of the chimeric drug, just let me, yeah. So to design a chimeric drug that contains the key pharmacophoric features of two different drugs. So basically chimeric drugs are the drugs which will have the key pharmacophoric features of two different drugs. So we are having two drugs. So every drug which have a particular pharmacophoric features means the features which are important for that drugs per activity, biological activity. So chimeric drugs are those drugs which will have the particular features of two or more different drugs in a single compound. So, for example, a structure containing the features of two methoxyestradiol and colchicine has been synthesized as a potent anti-cancer drug. So this is an example of a chimeric drug. Okay, we have two different drugs. One is 2-methoxyestradiol, which has this kind of steroid structure, and another we has a colchicine. So in colchicine, this trimethoxy ring or this group is very much important for this anti-cancer activity, and for this 2-methoxyestradiol, this bicyclic ring is very important. So by clubbing up these two things, we having this chimeric drug, which is a very potential anti-cancer agent. Although both of the parent structures have anti-cancer activity, they have serious drawbacks. 2-methoxyestradiol is metabolized quickly, while colchicine has the cytotoxic side effect. The chimeric structure has also anti-cancer activity with improved pharmacokinetics properties. So basically, this chimeric drug, which is a mixture, which is basically the pharmacophoric hybridization. So now this technique is known as the pharmacophoric hybridization, where we take up the important groups of pharmacophoric features from different drugs and club them up to derive a single compound which has the improved biological activity and improved efficacy, means improved pharmacokinetic effects. So this is a very good example of the chimeric drug, and the method which is used to for having this chimeric drug is known as the pharmacophoric hybridization. Okay, so number B is the correct answer. Now problem number 10, it says that 2 is found to be more interactive with the target rather than the parent lead compound Y. So we have a one parent lead compound, okay, here, and number 2 is the another compound which is more interactive than this parent lead compound 1. What is the correct reason? Your options are: number A, the strategy used was one of the ring expansion. Number B, the blue nitrogen allows better solubility. Number C, the blue nitrogen acts as a hydrogen bond donor. Number D, the strategy can be described as the extension since a further binding interaction has occurred. Which of the following is the correct statement regarding this phenomena, where it is found that this compound 2 is more active than the parent compound, lead compound 1? Your options are: number A, the strategy was used is one of the ring expansion. Number B, blue nitrogen allows better solubility. This blue nitrogen. Number C, blue nitrogen acts as a hydrogen bond donor. Number D, the strategy used could be described as the extension since one further binding interactions has occurred. So which of the following will be the correct answer for problem number 10? Okay, let me check the chat box now. Okay, so I have not received any answer for this particular question. Okay, it is fine. Let me just show you the answer and let us discuss about it. So the correct answer of problem number 10 will be, it is number D. The strategy used could be described as an extension since the further binding interaction has occurred. So basically, what is the extension of the structure? So as you can see, there is a nitrogen here from this compound number 2 from the lead compound 1. So because of this nitrogen, an extra binding interaction has occurred, and which is the reason for that this particular compound is more interactive than the lead compound. And the strategy used here for is known as the extension strategy. Okay, so extension of the structure. The strategy of extension involves the addition of another functional group or substituent to the lead compound in order to probe for extra binding interaction with the target. Lead compounds are capable of fitting the binding site and have necessary functional groups to interact with some of the important binding regions present. However, it is possible that they do not interact with all the binding regions available. For example, a lead compound may bind to 3 or more binding regions in the binding site but fail to use the fourth. Therefore, why not to add an extra functional groups to probe the further region? So this is an example. There is a drug. Okay, so let us say there is a drug and it has the receptor has 4 different interacting site, and the drug has only 3 interactions interacting groups which can interact with 3 of them but not the fourth one. So basically, what is the extension strategy is to use another functional group or any substituent on this position so that this drug can utilize the fourth interacting group also with the extra interaction. So this is known as the extension strategy. So extension tactics are often used to find extra hydrophobic regions in the binding site by adding various alkyl and aryl alkyl groups. These groups can be added to the functional groups such as alcohol, phenol, amines and carboxylic acid should they be present in the drug, as long as this does not disrupt the important binding interaction that are already present. Alternatively, they could be built onto the building blocks used in the synthesis of the various analogues by some token substituents containing polar functional groups could be also added to probe for an extra hydrogen bonding or ionic interactions. Okay, so this is the strategy of extension which is used for the designing of the drug. Now I will go to the next problem. Problem number 11 says Du1229290 is more potent than the selective than the saltopride because, so this is the saltopride, this compound, and this is the Du1229290. Okay, so this compound is found to be more active than this saltopride. What is the reason? Number A, pyrrole ring has made it more rigid. Number B, replacing amide moiety has improved the lipophilicity. Number C, both A and B are correct. And number D, it cannot be said for sure sure from only from the structures. Okay. So let me know A, B, C or D, which of the following is the correct answer for problem number 11. Number A says pyrrole ring has make this compound more rigid. Number D, replacing amide moiety has improved the lipophilicity. Number C says that A and B both are correct, and number D says that it cannot be said for sure only from the structure. So let me know which of the following will be the correct answer for problem number 11. Look at the structures very carefully, think and then let me know A, B, C or D, which of the following you think is the correct answer. For the last question, I have not received any answer, but for this one, I want all of you to participate and write any of the options you think is the correct answer. I have already mentioned it a couple of times that it does not matter even if your answer is wrong, but just try. Du1229290 is more potent and selective than saltopride. What is the reason? Let me check the chat box. Okay, I have got 3 answers. Very good. All of you have answered, and all of you are thinking it is number C. Okay, let us check if number C is the correct answer. The correct answer is number A. The pyrrole ring has made it more rigid. That is the only reason, because for this compound to be more active or and selective also. So basically, this pyrrole ring of Tu-122-290 serves to increase the rigidity of the side chain present in the saltopride. Okay, this is another example for the rigidification. This bond in the salto, this bond is okay. Here is not many marking. Just I will show you once with the pen. So basically this bond in saltoprime, okay, the in the is freely rotatable and it is locked within the pyrrole ring. So basically this ring is it can rotate freely in this case of the saltopride, but here it is locked with this pyrrole ring in the particular Du-122-290. This reduces the number of possible conformations that Du-122-290 can adopt and increases the chances of the active conformation being present when it enters the binding site, thus increasing the activity. Moreover, some of the conformations that are now disallowed may be those which are adopted when the saltopride binds with the D2 receptor, thus increasing the selectivity for the D3 receptor. Okay, so this is the pyrrole ring. This is the introduction of pyrrole ring as a bioisoster of the amphor and amide group. So basically, this pyrrole ring also is acting as a bioisoster of the amide group here. Okay. So answer problem number A was the correct answer. Actually, for this question, I will just recheck the answer one more time, okay? Because I think number C can also be correct. So this for this thing, I will recheck this particular thing because number A is the correct one. I'm not very sure about the number B, but now I'm thinking number B can also be a correct answer. But I will recheck this thing and update it in the next class, okay? So let us go to the problem number 12. So it says that which of the element acts as a bioisoster of hydrogen? Your options are number A, D, it means deuterium, number B, F means fluorine, number C, lithium, and number D, both A and B. Which of the following element or elements acts as the bioisoster of hydrogen? Deuterium, fluorine, lithium, or both deuterium and fluorine? Only deuterium, only fluorine, only lithium, or both deuterium and fluorine? Which of the following elements act as a bioisoster of proton? Think and let me know in the chat box. The element which acts as a bioisoster of proton, which will be the correct answer? What will be the correct answer? Let me check the chat box. A, B, and D. So I have received 3 different answers. One is thinking it is number A, another is thinking it is number D, and other also we have number B as the answer. Let me now check which of the following will be the correct answer. So the element which is the bioisoster of proton will be, it is number D, both A and B. Okay, so deuterium and fluorine, these elements both are the bioisoster of the proton or hydrogen. So basically, deuterium as an isostar of hydrogen. So the strategic development deployment of the deuterium at sites of metabolism where proton abstraction can disturb the metabolism and redirect the metabolic pathway. For example, here. So particularly here we have two protons in this position, right? So this is the paroxetine, okay, and this compound is inactive in CYP2D6. This is the side effect because this compound is toxic because of this proton which can be easily abstracted and which disturbs the metabolism of this drug. But this is the CTP341, which is the particular compound has only deuterium in place of the proton here. So this deuterated compound relieves the inactivation of CYP2D6, and these are the examples of the selective serotonin reuptake inhibitor which are used as the antidepressant drugs. Okay. Fluorine as an isostar of proton, the unique properties of fluorine have led to its widespread application in drug design as an isostar for proton, since the incorporation of fluorine can productively modulate a range of the properties of interest into medicinal chemist. Okay, so proton and fluorine both have the van der Waals radius around 1.20 and 1.35 respectively, which are almost same, and 20% of all drugs have the fluorinated compound. So fluorine is a very crucial element in the drug discovery because most of the drugs, as you can see, 20% of the all drugs are fluorinated compound, and fluorocompounds have a better activity. So one of the strategies to encounter the low metabolic stability is to block the metabolically liable site with a fluorine substituent and hope that the small fluorine atom will not impair the binding interaction with the target protein. Okay, so this is an example of the cholesterol absorption inhibitor or hyperlipidemia drug. Okay, so this is a compound which has a fluorine in this position and this position. So basically this para hydrogens. Okay, so here you can see there is a para group here. Okay, if you can say there is a bond and there is a proton which is a para to this particular group. Okay, so this position para protons are sometimes susceptible for the metabolism. So if we block this particular position with the fluorine, it will counter the low metabolic stability of that particular drug, and it will metabolically block this labile site. Also same is for this omethoxy group, which is very metabolically labile, which we can also remove it and introduce a fluorine here. So this is the lead compound which has a ED50 value of 2.2 milligram per kg, and this is the developed compound which has more metabolic stability, hence the ED50 value is reduced to 0.04 milligram per kg. Okay. So both fluorine and deuterium can be used as a bioisostar or isostar for the proton. Okay. Now problem number 13, it says which of the following statements regarding the lipophilic efficiency is false. Your options are number A, optimizing LIPE involved. LIPE means lipophilic efficiency. Okay. So number A, it says optimizing LIPE involved a parallel optimization of potency. Number B, drugs with a good level of the lipophilic efficiency will have a high hydrophobic character. Number C, drugs with a good level of lipophilic efficiency will have a low hydrophobic character. Number D, lipophilic efficiency can be calculated by the equation LIPE equal to PiC50 minus C log D. Which of the following statements regarding lipophilic efficiency is false? Again, I will repeat the statements. Number A says optimizing lipophilic efficiency involves a parallel optimization of potency. Number B, drugs with a good level of lipophilic efficiency will have a high hydrophobic character. Number C, drugs with a good lipophilic efficiency will have a low hydrophobic character. Number D, lipophilic efficiency can be calculated by the equation LIPE equal to PiC50 minus C log D. Which of the following will be the correct answer for problem number 13? Let me know in the chat box A, B, C or D which of the following is the correct answer for problem number 13. I will wait for 2 minutes and then I will check the answer and I will show you what will be the correct answer. Let me check the chat box now. Number C. Okay. I have received only one answer, which is saying it is number C. Let me check if number C is the correct answer or not. So the correct answer will be, it is number B. Drugs with a good lipophilic efficiency will have a high hydrophobic character. Now let us discuss it a little bit. So basically, this is one of the testing procedure where we measure the lipophilic efficiency. So when investigating the structure-activity relationships for the drug-target binding interaction, biological testing should involve in vitro tests. For example, inhibition studies on isolated enzymes or binding studies on membrane-bound receptors in whole cells. The results then show conclusively which binding groups are important in drug-target interaction. If in vivo testing is carried out, the results are less clear-cut, but the loss of activity may be due to the inability of the drug to reach its active target rather than reduce drug-target interactions. However, in vivo testing may reveal functional groups that are important in protecting or assisting the drug in its passage through the body. This would not be revealed by in vitro testing. NMR spectroscopy can also be used to test the structure-activity relationship. As mentioned in introduction, there is a little point of designing a drug that has optimum interaction with the target if it has undesirable pharmacokinetic properties. Calculating a structure's hydrophobicity can provide an indication as to whether the drug is likely to suffer from the pharmacokinetic problems. This is because the hydrophobic drugs have been found to be more prone to adverse pharmacokinetic properties. For example, they are more likely to interact with other protein ligands, resulting in unwanted side effects. They are generally less soluble, show poor permeability, and are more likely to produce toxic metabolites. The hydrophobic nature of the drug can be calculated by a C log D value. In recent years, several research groups have optimized drugs by optimizing lipophilic efficiency, or LIPE, where lipophilic efficiency equal to pKi or PIC50 minus C log D. Drugs with good level of lipophilic efficiency will have high activity and low hydrophobic character. Okay. So high activity means the pK1 value and PI50 value will be higher, and the low hydrophobic character. Optimizing LIPE involves the parallel optimization of potency and hydrophobicity. This quantitative method for optimizing both the pharmacodynamic and kinetic properties has been called the property-based drug design and was used in the structure-based drug design of crizotinib, which I have already mentioned earlier. Okay, so this optimization of this kind of pharmacodynamic and pharmacokinetic property is known also as the property-based drug design. So all the options, let us understand first one, which says lipophilic, optimizing the lipophilic efficiency involve parallel optimization of potency, which is correct. Drugs with good lipophilic efficiency will have a high hydrophobic character. It is incorrect, as we have already seen that the drugs will have a low hydrophobic, good level of lipophilic efficiency will have low hydrophobic character and high activity. Okay, means the higher value of PIC50 value. And this can be calculated by the equation, which is also correct. So problem number B is the statement is a false for this regarding the lipophilic efficiency. Okay. So now I will go to the next problem. So problem number 14 says, which of the following option is correct? You have an equation where this compound is converted to A and then B. So now you have to identify which of the following is the correct answer. A equal to this one, B equal to this one, A equal to this, or B equal to this. Which of the following will be the correct answer? Okay, sorry for this. I just one second. So I think you have already seen the answer because I just forgot to give the animation. Okay, it's fine. Just let me correct it. So I just forgot to put the animation here. Okay, it's fine. Just let me show you the answer then. I think you have already seen the answer for problem number 14. Just forgot to put the animation there. Okay, it's fine. So the correct answer for this will be number B. So this is the equation. Okay, so this is basically the dealkylation of the tertiary amine and formation of the secondary amide. So this is the tertiary amine. Okay, and first this reaction with the dealkylation or demethylation. Okay, so this is the removing this methyl group and introducing a proton here, and followed by the CH3COCl, which give rise to the secondary amide group. Okay, so basically the important thing is what are the basically binding modes for this amide. So basically here we have this primary amide. So as you can see, so amides are very important functional group in our biological system, right? Because we have, as you remember, we have our peptide bonds, which are a type of the amide linkage is there, and also on. So basically, primary amides have this four different interaction is possible. Well, the two are the hydrogen bond donor from this amide group, and hydrogen bond acceptor is the ketonic group. Okay, so as we are changing the primary amide to secondary amide, the hydrogen bonding accepting is the same, but the donor is decreasing from primary to secondary and tertiary. The tertiary amides have only hydrogen bond acceptors, but not in the donors, right? So binding role of the amides, many of the lead compounds correctly studied in the medicinal chemistry are the peptides and polypeptides consisting of amino acids are linked together by a peptide or amide bonds. Amide are likely to interact with the binding sites to the hydrogen bonding. The carbonyl group, carbonyl oxygen atom can act as a hydrogen bond acceptor and has the potential to form two hydrogen bonds. Both of the lone pairs involved are in the sp2 hybridized orbital, which are located in the same plane as the amide group. The nitrogen cannot act as a hydrogen bond acceptor because the lone pair interacts with the neighboring carbonyl group as I described earlier. Primary and secondary amides have a NH group, which allows it possible for this group to act as a hydrogen bond donor. Okay. Let us directly go to the problem number 15. So it says that the enzyme which is responsible for the conversion of estrone to the female steroid hormone estradiol is number A NADH, number B beta hydrogenase, number C beta hydroxysteroid dehydrogenase, and number D alpha hydroxysteroid dehydrogenase. Which of the following enzyme is responsible for conversion of estrone to the female steroid hormone estradiol? NADH, beta hydrogenase, beta hydroxysteroid dehydrogenase, or alpha hydroxysteroid dehydrogenase. Which enzyme is responsible for the conversion of estrone to estradiol? Let me know A, B, C, or D. Which of the following you think is the correct answer? Let me check the chat box now. I have received only one answer, which is saying it is number C. Okay, let me check if number C is the correct answer or there is something else. So the enzyme which is responsible for the conversion of estrone to estradiol will be, yes, it is number C, beta hydroxysteroid dehydrogenase. So converting an enzyme substrate to an inhibitor by the extension tactic. So the enzyme 17 beta hydroxysteroid dehydrogenase type 1 catalyzes the conversion of the estrone to the female steroid hormone estradiol with the cofactor NADH acting as the reducing agent for the reaction. Okay, so inhibition of this enzyme may prove useful in the treatment of the estradiol dependent tumors as the levels of the estradiol present in the body would be lowered. The cofactor NADH is a cofactor. Okay, so first of all, the NADH is bound to the next to the estrone to an active site, and so it was reason that a direct bonding interaction between an estrone analog and NADH would lock the analog into the active site and block the access of the estrone itself. Therefore, the analog would act as an enzyme inhibitor. Various substituents were added to the position of number 16 to achieve this goal. To crystallographic or molecular study has shown that substituent would be ideally placed for an interaction with the cofactor. This led to the structure number 2, which is the which is the number which showed so this is the structure which showed the promising activity of an inhibitor. The amide group interacts with the primary amide NADH by hydrogen bonding, while the pyridine ring interacts with the phosphate groups to the cofactor. A more conventional extension strategy was to add an ethyl group at C2, which allowed the additional van der Waals interaction with the small hydrophobic pocket of the active site. It was also observed that two protons acted as steric blockers, prevented NADH, reducing the ketone group of the analog. So basically, this is the estrone to the estradiol. Okay, and this is the enzymes and how this enzyme catalyzed conversion happens, and this is the conversion of the enzyme substrate to an inhibitor by this extension strategies. Okay, extension strategy I have already discussed. What is the extension strategy? Is to introduce a more functional group to introduce extra bonding interaction. So this is the thing. Okay, so the option number C was the correct. That is the enzyme 17 beta hydroxysteroid dehydrogenase type 1 catalyzes this particular conversion, and cofactor NADH is used here as the reducing agent. Okay. Now let me go to the next problem. Problem number 16 says which of the following statements is true regarding isostars? Number A, isostars are the atoms or group of the atoms or the groups of the atoms which share the same valency but have different chemical and physical properties. Number B, replacement of OCH2 with SCH2 retain the activity of the beta blocker propanolol. Number C, replacement of the OCH2 with NHCH2 eliminates the activity of the beta blocker propanolol. Number D, isosteric groups can be used to determine whether a group is involved in hydrogen bonding. Which of the following statements is true regarding the isostars? Number A, isostars are atoms or group of atoms which share same valency but have different chemical or physical properties. Number B, replacement of OCH2 with SCH2 retains the activity of a beta blocker propanolol. Number C, replacement of OCH2 with NHCH2 eliminates the activity of the beta blocker propanolol. And number D, isosteric groups can be used to determine whether a group is involved in hydrogen bonding. Identify the correct statement. Which of the following statement will be true regarding the isostars? I will give you a minute, then I will check the chat box and we will discuss it in detail. Let me check the chat box. Okay, I think I don't have any answer for that question. Okay, it's fine. Let us check which of the following will be the correct answer. So the statement which is true, number D, isosteric groups can be used to determine whether a group is involved in the hydrogen bonding or not. So isostars are the atoms or the atoms or groups of atoms that which share the same valency and which have the same chemical and physical similarities. For example, SH, NH2, CH3 are the isostars of OH, whereas S, NH and CH2 are the isostars of O. Isostars can be used to determine whether a particular group is an important binding group or not by altering the character of the molecule in the controlled way of possible. Replacing O with CH2, for example, makes a little difference in the size of the analog, but it will have a marked effect on the polarity, electronic distribution, and bonding. Replacing OH with larger SH may not have such an influence on the electronic character, but the steric factors become more significant. Isosteric groups could be used to determine whether a particular group is involved in hydrogen bonding. For example, replacing OH with CH3 would completely eliminate the hydrogen bonding, whereas replacing OH with the NH2 would not. The beta blocker propanolol has the ether linkage. Replacement of the OH-CH2 segment with the isostar CH1CH SCH2 CH2CH2 eliminates the activity, whereas the replacement of NHCH2 retains the activity, though reduced. This shows that an ether oxygen is important for the activity of the drug and suggests it is involved in the hydrogen bonding with the receptor. Okay. So, bioisostars are the substituents of the group which has similar physical chemical properties which produce the broadly similar biological properties of the chemical compound. In drug design, the purpose of exchanging one bioisostar to another is to enhance the desired biological and physical properties of the compound without making significant change in the chemical structure. Okay, for example, this is the aminopyridine, which is marketed as the analgesic and anti-inflammatory drug in 1896. In 1922, it was revealed that it was a carcinogenic. It is a carcinogenic compound. Okay, so biogenetic modification happens when we change this here this amine, tertiary amine with a normal alkyl chain, and it is developed by Roche in 1951. The bioisostatic modification of dimethyl amino group removed its carcinogenic effect. Okay. So this is the example how bioisostatic modification improves, enhance the activity or eliminates any undesired activity. Okay. So now I will go to the next problem. Problem number 70 says introducing a methyl substituent to the dopamine antagonist results in a dramatic reduction in affinity. What is the role of the methyl group? Number A, metabolic blocker. Number B, configurational blocker. Number C, conformational blocker. Number D, beta blocker. Introducing a methyl group to the dopamine antagonist results in a dramatic reduction in the affinity. What is the role of the methyl group? Metabolic blocker, configurational blocker, conformational blocker, or beta blocker? Let me know in A, B, C, or D which of the following you think is the correct answer. Let me check the chat box. Okay, no answer. Okay, it's fine. Let us discuss it. The correct answer will be number C, conformational blocker. Okay. So what are the conformational blockers? So this is the dopamine, your particular dopamine antagonist, which I was referring to. Okay. So another tactic that has been same effect to use the conformational blocker in certain situation, a quite simple substituent can hinder the free rotation of a single bond. So we have earlier have discussed about the rigidification where the introducing a double bond hinders the rotation of the free bonds or introducing a I mean introduction of a group, a particular ring also hinders the rotation. Another one is to introduce a simple substituent like methyl, which can also hinder the free rotation of a single bond. Introducing a methyl substituent to the dopamine D3 antagonist gives you the structure 2, this one. Okay, here there is a only interaction of a methyl group and this results in the dramatic reduction of the affinity. The structure 1, the free rotation around this bond happens and this allows the molecule to adopt a conformation, whereas the aromatic ring are the coplanar or the active conformation of the receptor. Okay, in this case the conformation blocker rejects the active conformation. In other cases, to increase activity is also possible. The steric clash between the new methyl group and the orthoproton of the neighboring ring prevents the both of the rings to be on the same plane. Free rotation around the bond between two rings are no longer possible, and so the structure adopts a conformation where two rings are at the angle of each other. And because of that, this particular compound activity is lessened, because they, for the adopting a conformation, active conformation, the four ring should be in on same plane. But due to the introduction of the methyl group here, the free rotation is not possible, and now these two rings will not be on the same plane. It will be on a different planes. And this methyl group here is acting as a conformation blocker because it is hindering this molecule to adopt the active conformation which is required for the optimum activity. Okay, so number C is the correct answer. Now, and we'll go to the next problem. Problem number 18 says, which of the following statements is false regarding the lactams? Your options are number A, they act as an acylating agent and irreversibly inhibits the bacterial enzyme by acylating the serine residue on in the active site. Number B, they can form intermolecular hydrogen bonding. Number C, the lactam can undergo chemical reaction with the target if the ring size is large and no strain is suffered. Number D, a prominent example of lactam ring is present in penicillin. Which of the following statements is false regarding lactams? Your options are number A, they act as an acylating agent and irreversibly inhibits bacterial enzyme by acylating the serine residue in the active site. Number B, they can form intermolecular hydrogen bonding. Number C, the lactam can undergo chemical reaction with the target if the ring size is large and no strain is suffered. Number D, a prominent example of lactam ring is present in penicillin. Which of the following statements is false regarding lactams? Which of the following statements is false regarding lactams? Okay, let me check the chat box. I have one answer which is saying it is number A. Okay, let me check if number A is the correct answer or not. So the correct answer will be, it is number C. The lactam can undergo chemical reaction with the target if the ring site is large and no strain is suffered. This statement is not correct. Okay. So Basically, amides that are within a ring system are called the lactams. They too can form the intermolecular hydrogen bond. However, if the ring is small and suffers ring strain, okay, the lactam can undergo chemical reaction with the target, leading to the formation of the covalent bond. And the best examples are the penicillin, which acts as a acylating agent, irreversibly inhibits the bacterial enzyme by acylating serine residue of the active site. Okay, so lactam can undergo the reaction if the ring size is small and there is a ring strain. Only then this particular lactam ring can undergo with the chemical reaction of the target. That is why the statement number C is not correct. Okay, other statements, first one which says that it acts as acylating agent and irreversibly inhibits the bacterial enzyme by acylating serine residue. Is correct. It can form intermolecular hydrogen bonding. It is also correct. And the prominent example is penicillin, where the lactam ring is present. Okay, so basically, all penicillin contain a four-member beta-lactam ring. This ring contains two carbon atoms that are sp3 hybridized, and nitrogen atom that is sp3 hybridized, and a carbon atom that is sp2 hybridized. The structure is similar to the combination of two amino acids, cysteine and valine. Because of the restrictions of the ring, the normal bond angles are 109.5 and 120 are not able to obtain, so the ring is strained. This mixed amide is highly reactive. The ring opens up to the penicillin, can become covalently bonded to the enzyme and synthesize the cell walls of the bacterium, thus blocking the action. Okay, so if the ring size is large and no strain is there, the lactams are not active. They will not undergo the chemical reaction. But if it has a small ring size, so that and there is a ring strain, it will be highly active and it will react. Okay, so the statement number C was the false one. Now, problem number 19, it says which of the following statement is false regarding the binding role of alkenes? Number A, they are flat and hydrophobic. Number B, they like aromatic rings can interact through the van der Waals interaction. Number C, they interact with the binding site less poorly compared to their saturated analogues. Number D, they are easier to reduce compared to aromatic rings. Which of the following statements are false regarding the binding role of the alkenes? Number A, they are flat and hydrophobic. Number B, they are like aromatic rings can interact through the van der Waals interaction. Number C, they interact with the binding site less poorly compared to their saturated analogues. And number D, they are easier to reduce compared to the aromatic rings. Which of the following statements is correct? Means which of the following statement is false regarding the binding role of alkene? Let me know which of the following you think is the correct answer for problem number 19 in the chat box, A, B, C or D. Which option is the correct one? Which statement is false regarding the binding role of alkene? Number A, they are flat and hydrophobic. Number B, they are like aromatic ring can interact through van der Waals interaction. Number C, they interact with the binding site less poorly compared to their saturated analogues. Number D, they are easier to reduce compared to the aromatic rings. Okay, let me check the chat box. Okay, there is no answer for this problem number 19. Okay, it's fine. Let me check which of the following will be the correct answer. So the statement which is false regarding the binding role of the alkenes will be, it is number C. They interact with the binding site less poorly compared to their saturated analogues. Okay. So like aromatic rings, alkenes are planar and hydrophobic, so they can too interact with the hydrophobic regions of the binding site through the van der Waals interactions. The activity of the equivalent to the saturated analog would be Vogt testing, as the saturated alkyne region is bulkier, cannot approach the relevant region of the binding site closely. Alkenes are generally easier to reduce than the aromatic ring, so it may be possible to prepare saturated analogs directly from the lead compound. So they actually, in case of the what happens in case of the alkenes, they have the flat ring and the hydrophobic region. They can form the van der Waals interaction, which is not present in case of the saturated one. In the case of saturated rings, the hydrophobic interaction is very less, so that is why they interact more. The saturated analogs interact more poorly compared to the alkene ones. So number 6 statement which says they interact with the binding site less poorly compared to the saturated analog, it is false. Okay, with the alkenes they interact more. The saturated analogs they interact poorly rather than the our alkenes. Now I will go to the last question of today's session. So the last question of today's session, problem number 20, says which of the following statements is false regarding the quaternary ammonium group? Your options are: number A, the neurotransmitter acetylcholine contains a quaternary ammonium group. Number B, quaternary ammonium salts interact with the carboxylate group through the hydrophobic interaction. Number C, quaternary ammonium salts interact with the phenyl rings of the binding site through the induced dipole interaction. Number D, none of the above. Problem number 20, it says that which of the following statements is false regarding the quaternary ammonium group? Your options are: number A, the neurotransmitter acetylcholine contains a quaternary ammonium group. Number B, quaternary ammonium salts interact with the high carboxylate group through the hydrophobic interaction. Number C, quaternary ammonium salts interact with the phenyl rings of the binding site through the induced dipole interaction. Number D, none of the above. Which of the statement is false? Let me know in the chat box A, B, C or D, which of the following will be the statement is false regarding quaternary ammonium group. I will wait for a minute, then I will check the chat box. Okay, let me check which of if there is any answer in the chat box. Okay, it's fine. Let me show you which of the following will be the correct answer. So the statement which is false regarding the quaternary ammonium group is number B, which says that quaternary ammonium salts interact with the carboxylate group through the hydrophobic interaction. This statement is false. So basically, binding of the quaternary ammonium salts are ionized and can interact with the carboxylate groups by ionic interactions. Another possibility of an induced dipole interaction between quaternary ammonium ion and its aromatic ring at the binding site. The positively charged nitrogen can distort the pi electron of the aromatic ring, such as the dipole is induced, and whereby the face of the ring is slightly negative and the edges are slightly positive. This allows the interaction between the slightly negative faces of the aromatic ring with the positive charge of the quaternary ammonium ion. This is also known as the pi-cation interaction. The importance of these interactions could be tested by synthesizing an analog that has a tertiary amine group rather than the quaternary ammonium group. Of course, it is possible of such groups can ionize by becoming protonated and then interact with the same way. Converting the amine to the amide would prevent this possibility. The neurotransmitter acetylcholine has a quaternary ammonium group which is thought to bind to the binding site of its target receptor by the ionic bonding or induced dipole interaction. Okay, so statement number 1, it says neurotransmitter acetylcholine has the quaternary ammonium group. It is the correct one. Number B, which says quaternary ammonium salts interact with the carboxylate group through the hydrophobic interaction, it is not correct. But it does statement number C, which says quaternary ammonium salt interacts with phenyl ring with the induced dipole interaction, it is correct. Okay, the number B is incorrect because there is no hydrophobic interaction, rather it interacts with the carboxylate group with the ionic interaction. So as you can see here, Whenever the drug has a quaternary ammonium ion, it will interact with the carboxylate ion with this kind of plus negative positive in ionic interaction. But in case of the phenyl ring, there will be an induced dipole in the ring, whereas the pi electron cloud will acting as a negative dipole, and the edges of the ring will act as a positive dipole, and there is the induced dipole interaction with the phenyl ring. Okay, so so option number B was the correct answer. So this was the last question of today's session. So today what we discussed mainly, we have discussed regarding the different strategies of the optimizing of a drug and target interaction. We have discussed about rigidification, extension strategy, simplification, and all this conformation blockers, then bioisostars and ring strain, how it behaves, all the functional groups. That is why which we have discussed today. So I am now sharing my email ID with you. You can just let me know. 1 second. So this is my email ID. If you have any doubt, any query regarding today's session or any other our previous session, you can let me know through this email ID. Okay, so today it was our week 8 live session, and on next Friday from the same time from 4 to 6 p.m. we will have our next live session which where we will discuss the week 9 course content. That will be our 9th live session. Every week we are having this kind of live session, and at the end of our course we will have a summary session of around 1 to 2 hours where I will summarize the complete course content. So if you have any doubt or any query, just let me know through the email. I will try to answer those query in the our next class, or also we will have a summary class at the end of the session where you can just drop your queries beforehand, and I will discuss that topic. Okay. I have already shown you where to find the problem solving section. You just have to go to the NPTEL website and go click on the problem solving recording. Then you will redirect it to a page where we will find this today's class PPT and also the recordings. So if there is no issue, I will close the session for today. Okay, thank you all for joining. I am closing the session for today. Let's meet on the next Friday. Meanwhile, if you go through the slides, go through the lecture PPT and let me know if you have any doubt or any query. Thank you all. I am closing the session for today.